Hello friend, all are welcome to my channel GetEasyMedicalMicro. Today we will discuss about the bacterial capsule and slime layer. So what is bacterial capsule? Many bacteria secrete extracellular polymerase substances in natural environment. When it is closely attached or condensed around the bacterial cell wall, it is called capsule. So what is this? Suppose this one is bacteria like this, and this one is bacterial cell wall. It is closely attached with the cytoplasm membrane. Okay. When another extra layer it is a polymerase substance, polymer like nature, and it is secret from its own cell structure like this. This one is secreted from own cell structure, and it is easily get attached with its body. This is called capsule. Okay, this is called capsule. Again, I am telling you, many bacteria secrete extracellular polymerase substances in natural environment. When it is closely attached with the or completely condensed with the outer layer of the bacterial cell wall, it is called bacterial capsule. Okay, this one is capsule. Okay, now it has two types. It's polypeptide and polysaccharide in nature, and we are discussing the another part also, slime layer. What is slime layer? These extracellular polymerase substances, when secreted from the bacterial cell wall, it is not attached with the bacterial cell wall. Okay, that time this nature is called the slime layer. Okay, so again I am telling you, slime layer is the same thing. These polymerase substances secreted from the bacterial cell wall, but it not get easily attached with the outer part of the bacterial cell wall. It is called slime layer. Now we are giving the example leuconostoc slime layer, and the capsule containing bacteria is, we all know, pneumococcus, streptococcus pneumoniae, pneumococcus. So now comes to the function of the capsule. So what is the function? If capsule is present, then it also providing extra protection in bacterial cell wall. One protection, and also it provide the protection of cytoplasmic membrane. Okay, so now comes to the how detection of capsule. Detection of capsule. There are generally 4 methods. One is gram stain. Number 2 is negative staining by Indian ink. Number 3, copper salt as a mordant. Number 4, electron microscopy. These 4 methods are there, and also one method is present, that is the serological reaction, and it is very rare reaction. Okay, it is very rare, we generally perform. So this one is very rare. Okay, so we will discuss one by one. So first come to gram stain. We already discussed in my previous lecture gram staining procedure. So in gram stain, generally bacterial capsule is not properly visible. In case of Klebsiella pneumoniae and so many other bacteria present, after gram staining procedure, when we will observe in under the microscope, that time will visible little bit, and we look the bacteria will be broad in shape. Okay, that time we can realized the bacteria is capsule containing bacteria. Okay, now second is the negative stain by Indian ink. Okay, Indian ink as a negative stain when the bacterial sample mixed with Indian ink and we observe under the microscope, we seen a clear capsule like this with a dark background like this. If we see the Streptococcus pneumoniae, we see like this. So this one is capsule containing bacteria, look like this against the dark background. Against the dark, we will see this one is looking like this. Okay, and we will observe under dark background, and this total portion, that time we will definitely say this one is capsule containing the bacteria. This one is showing capsule against dark background. Okay, that is the negative stain of Indian solution. Next one is number 3 is copper salt as a mordant. Okay, when we will performing the gram stain procedure, that time we using mordant as a iodine, grams iodine. Against of grams iodine, if we use the copper salt, that time the copper salt easily attached with the bacterial cell wall or primary stain. Okay, then it make a purulent cover, means extra cover. Okay, so as well the capsule is basic in nature. Okay, the capsule or extracellular polymerase substance, the basic in nature. When copper salt using as a mordant, that time it will closely attached with over the cell wall. And after preparation of gram staining, we are using as a copper salt as a mordant. That time the bacteria will swim broad in nature or bigger in size at the time of microscopy. So that is the one of the procedure. Okay. Next is very easy for electron microscope. Electron microscope. So we know electron means very little amount, means micron, very micron amount we can observe easily under this electron microscope. We know this one is two type of electron microscope. One is TEM, another one is SEM. Transmission electron microscope and scanning electron microscope. Transmission means we can observe the capsule on TEM, transmission electron microscope. Okay, so we can easily observe under the microscope and also visible the clear structure in the bacteria containing capsule like this. Okay, next the another one is, I said, the serological reaction. The serological reaction. Serological reaction. And it is also called the Qualling reaction. Quellung. Qualling reaction. What is this? Qualling reaction, it is also called the anti-capsular antibody. So this capsule, this is a polypeptide or polysaccharide in nature or polymer like this. So it act as an antigen when it enter inside our body. Okay, so it act as a capsule, act as an antigen. So if antigen, suppose this one is capsule, this one is capsule, okay, this one is bacteria. Okay, this one is bacteria like this. Okay, this one is capsule. It act as an antigen when enter inside our body. If this antigen is treated with specific antibody against this capsular antigen. So specific antibody, it should be in specific antibody. If the specific antibody treated with that specific capsular antigen, that time the antigen antibody reaction will happen and make a broad in shape or bigger in shape. Like this one is, suppose it is very easy to understand. If okay, if antibody is treated with this antigen, that time suppose this one is antibody, okay, okay, so it easily attached with the capsular antigen and form a broad or bigger in shape. When we will observe under the microscope, that time the capsule containing bacteria is bigger in size. Okay, so that is the main fundamental things. If it will be very clearly if this one is like that. Okay, so this is the main fundamental things. So if capsular antibody treated with capsular antigen and which will be specific in nature, that time it will cause capsular antigen antibody reaction and make a bigger in size. At the time of microscopic examination, we will observe the capsule containing bacteria is bigger in size. So that is the another method, serological method. We can easily detect the capsule of bacteria. Okay, so this much is helpful for you. Thank you.